Welcome to ClinMicro Simplified, where microbiology becomes clear, simple, and a little bit fun. Let's dive into today's topic. Influenza is one of the few viruses for which the vaccine formula changes almost every year. This is not because scientists are confused, but because the virus itself keeps changing. To understand this, we need to understand how influenza behaves inside the human body and in the global population. Influenza is an RNA virus. RNA viruses replicate fast, but they make mistakes while copying their genetic material. Unlike DNA viruses, influenza lacks an effective proofreading system. These small errors slowly change the virus over time, especially the parts our immune system recognizes. Our immune system mainly recognizes influenza using two surface proteins, hemagglutinin and neuraminidase. Antibodies formed after infection or vaccination are directed against these proteins. If these proteins change, old antibodies stop working effectively. The most important reason the influenza vaccine changes yearly is something called antigenic drift. This refers to small, continuous mutations in hemagglutinin and neuraminidase. Even tiny structural changes are enough to help the virus escape previously formed antibodies. When antigenic drift occurs, the virus no longer looks exactly like the one used in last year's vaccine. So even if antibodies are present, they bind weakly or not at all. This is why last year's vaccine cannot fully protect against this year's circulating strains. Influenza is monitored globally through a WHO coordinated surveillance network. Thousands of virus samples are collected from different countries. Scientists track which strains are circulating, spreading fastest, and causing the most disease. Based on global data, experts predict which influenza strains are most likely to circulate in the upcoming season. The vaccine is updated accordingly, usually including 3 or 4 strains. This decision is made months in advance because vaccine production takes time. Sometimes the virus continues to mutate even after the vaccine strains are selected. If a major drift occurs later, vaccine effectiveness may be lower. This does not mean the vaccine failed. It still reduces severity, hospitalizations, and complications. This yearly change is most prominent in influenza A viruses. Influenza B also mutates, but more slowly. Many other viruses are genetically stable, which is why their vaccines last for years or even a lifetime. So the influenza vaccine changes every year because the virus itself keeps changing. Antigenic drift alters the surface proteins, making old immunity less effective. Updating the vaccine is the best way to stay one step ahead of the virus. If antigenic drift did not occur in influenza viruses, how would this change our global influenza vaccination strategy? Think about this scenario and share your answer in the comment section. I would love to see how you connect immunology with public health here. Do like, share, and subscribe for more simplified clinical microbiology content. Thank you for watching. I will see you in the next video.